Good evening, everyone. On behalf of myself and my co-laureates, Professor Steve Wilhelm and Mike McKay, I would like to sincerely thank the Daylight Academy for this prestigious award. It is an honor and a privilege to stand before you all here today and to have our research recognized here on this international stage. We are deeply grateful for this recognition and for the opportunity to contribute to the advancement of our scientific understanding of the importance of daylight. My name is Britt Zepernick, and I, like my co-laureates, am classically trained as an aquatic microbiologist. Now, you may find yourself wondering, why do aquatic scientists who study light beneath the surface of our lakes and our oceans care so much about daylight? If I had to answer this question very briefly, I would say daylight is the driving force and the currency of nearly all life on Earth. Almost all the energy on the planet originates from the sun, and biology is constantly evolving new and creative ways to harvest this daylight, this energy, and convert it into life. Seen here on this slide is an image I took in 2023 on a research expedition on Lake Erie of the North American Great Lakes. And we can visualize daylight streaming down and shining onto the surface of the water column. And beneath that surface are billions, if not trillions, of microscopic photosynthetic organisms called algae, or phytoplankton, that are capturing and harvesting this daylight. And these phytoplankton are using this daylight in ways that confer large-scale, truly global benefits. About 50% of all photosynthesis on the planet is performed within the aquatic water column, within our lakes and our oceans. This means that roughly 50% of our global carbon dioxide is consumed by these phytoplankton, and 50% of our atmospheric oxygen is produced in tandem with carbohydrates and sugars. To sum it up, life is literally created out of thin air. And when we think about the mechanisms by which algae are harvesting this daylight, some of us might picture something along the image to the left here, the photosynthetic apparatus that some of us learned in our introductory biology course. We've been studying this now as aquatic scientists and as photosynthetic biologists, as daylight researchers, for about 250 years. Yet, it was only recently that we started to uncover a variety of mechanisms that organisms use to capture this daylight that are distinct from photosynthesis. Mechanisms that were largely ignored and unexplored within our freshwater ecosystems. One of those alternative mechanisms is pictured here to the right, rhodopsins. Rhodopsins use the pigment retinol in place of chlorophyll A to harvest daylight and maintain cellular energetic quotas during times of stress when photosynthesis may be otherwise limited. Historically, us aquatic scientists like to study the algae in the water column and their mechanisms for harvesting daylight in the summer, when we assumed algae to be most abundant, due in part to warm temperatures and the sheer magnitude of daylight that was available. Consequently, the winter water column was thought to be a period of dormancy, of darkness, devoid of life. My colleagues and I wanted to ask the question, is there algal life hidden beneath the surface of ice? And does daylight play a role in shaping that algal life, that physiology? To investigate this question, our team embarked on a 2007 expedition to sample the winter water column of Lake Erie. For those of you doing the mental math in the room, no, I was not a part of this expedition. In 2007, I was 11 years old. But my co-laureates were aboard this mission, and what they found was a winter water column teeming with life. That coloration that you see, that brown, That's not mud, that's not sediment. Those are billions of algal cells that are specially adapted to not just persist, but thrive beneath that ice cover. What we found was that these algae are just as photosynthetically active as the summer algae in Lake Erie. And what's more, they play a fundamental role within this ecosystem. Most of the algae in this image are diatoms, or as I like to call them, master architects. As diatoms grow, they build these specialized silica or glass skeletons. And we can visualize these silica skeletons using specialized fluorescent dyes and microscopy, like in the image to the bottom left here. What's more, we can degrade and remove their pigments and a lot of the biological material from these diatoms. And all that remains are their silica skeletons, as my colleague Dr. Tricia Sponborough has done in these images to the right. Early work from our team deduced that these winter diatom communities play a fundamental role in supporting the winter food web and ecosystem in Lake Erie. In turn, this means that they also support the billion-dollar Great Lakes fisheries industry, rendering them of critical importance. This also, though, begs the question: how might these winter diatom communities, which are specially adapted to thrive beneath the ice cover, fare in a future water column where ice cover is drastically reduced? It's no secret that ice cover is disappearing across the Great Lakes region, and also in temperate freshwater systems across the globe. We can visualize some of this data shown here on this slide. This is data from Lake Erie's historical ice cover. Lake Erie, an abnormally shallow lake, has seen an increase in the frequency of low ice cover years, specifically throughout the past 3 decades. We can further then visualize this in real time using satellite imagery. And by looking at the picture to the left, in the bottom we have our ice-covered Lake Erie, and comparing it to the image to the right, our ice-free Lake Erie, we can infer that the presence or the absence of ice cover has substantial biotic and biogeochemical implications for those algae residing within this winter water column. Prior work from our team hypothesized that these winter diatom communities may potentially be starving for daylight within the ice-free water column, and this work was previously published in the Environmental Microbiology Reports Journal. What was suggested was that in the absence of ice cover, and due in part to the shallow nature of Lake Erie and the prevailing westerly winds which sweep across this region, we would see the mixture of our Lake Erie winter water column into this soupy, sedimenty mixture, and perhaps diatoms might be starving for daylight as a result. To investigate this, our team established a winter molecular time series of Lake Erie. And for those doing the mental math, this is where I came into play and joined the team for my PhD. We used an emerging tool called metatranscriptomics, which allows us to look at all the gene products across an environmental community and deduce what those organisms were trying to do at the exact moment that we sampled those from the water column. Historically, aquatic scientists largely looked at the DNA of organisms via a process called genomics. When I explain this in the classroom to my students, I like to use an analogy. Genomics is looking at all the tools that an organism could be using. It is the equivalent of peering in and looking through someone's toolbox and all the tools that they might possess. Transcriptomics is the process of looking at those active genes and their gene products. So if we continue the analogy, that'd be the equivalent of peeking in on an individual and noting that out of all the tools in their toolbox, they were using a hammer to build something at that exact moment. We wanted to deduce what our winter diatom communities were doing in the ice-covered versus the ice-free water column. And so to do so, we performed transcriptomics. And what we found was that our diatoms we're using a very different mechanism to harvest daylight within the ice-free winter water column. These ice-free conditions, might I add, which are going to be increasingly prevalent during present to future scenarios. We can start to the left of this diagram, a rendition of our historical Lake Erie, covered in ice. And we found that the diatoms collected from these conditions were trying to perform photosynthesis, using that as a means to harvest this daylight, where they situate themselves conveniently directly beneath the ice cover, where they can maximize their location in the water column to harvest this daylight. In contrast, to the right, we have our ice-free Lake Erie, where in the absence of this ice cover, we have the mixing up of our shallow lake into this soupy sediment mixture, a mixture where daylight is decreased and its spectra is shifted. Here we found evidence that these diatoms were trying to use rhodopsins as an alternative mechanism for harvesting daylight. We further found evidence that they were attempting to raft together via the expression of genes called fasciculins, in a means to potentially become buoyant and float to the surface of the lake water column, where they could evade light limitation and harvest that daylight they so desperately need. In total, this work suggests that while the climate change is shifting the light quality and availability within our winter water column, algae are evolving and adapting creative ways to harvest this daylight. To sum it up, daylight is the currency of life within our winter water column, but algae have really creative ways to tug on the purse strings, making sure that this energy currency is available not just to them, but to the broader food web and to individuals, including yourself and I. If you're interested in learning more about this work, I encourage you to read our piece in the conversation, and we thank the editors of this journal for working with us to render this research accessible to the broader community. We also want to thank and recognize our colleagues and our co-authors who played a role in ensuring that this work was produced via 2 publications, the ISME Journal and ASM Microbiology Resource Announcements. We also want to thank the US and Canadian Coast Guards for helping us collect these precious samples, and our funding resources, the Joint Genome Institute, NSERC with Canada, and the NSF and NIH with the US, who support the Great Lakes Center for Freshwater and Human Health. I want to end with a quote that I think illuminates some of the work that I shared with you today, and also on a personal level, it captures the scientific process to me and the pursuit of knowledge as we try to uncover the secrets of daylight. Lastly, I would like to thank the Daylight Academy and you all for your attention and for being here today to celebrate daylight. Thank you.